All right. So this section is actually, I thought it was just 4.4, but again, it's a new book and I'm still waiting for my new book. I had a feeling I'm not going to get it unless I ask because, I mean, I have the virtual one. We have some turnover going on in the main office, so I have to inquire again. All right. So this actually, there's two components to this. This is the bulk or the main content of this chapter is 4.4. We're going to talk about eukaryotic cell organelles. And then 4.5 talks about organelles found in plant cells. Okay, so it's like the plant cells have all of these. Their plant cells are eukaryotic, but plant cells have some special organelles as on top of what we're going to talk about. So they're in 4.5. So we're going to do that all today. So we're going to start with the endomembrane system. The endomembrane system consists of all of these things: the nucleus, the ER, the little vesicles here, the Golgi, some more vesicles, some lysosomes, and then the plasma membrane. So all of this is referred to as the endomembrane space or endomembrane system. So the nucleus, my face up here, the nucleus contains the cell's DNA. I hate that when people, when I ask, well, what's the function? What's the job of the nucleus? Everybody's like, oh, it's the control center. No, the DNA is the control center. The nucleus's job is to protect the control center. It protects the information that's inside here, which is the DNA. We have this really large, we have this double membrane here. It folds over on itself. For the nuclear membrane. Like I said, it separates the DNA from the cytoplasm. It has these big pores here, these pores, and we need those pores because we're going to have big things moving outside of the nucleus. And so we do have some things moving in, but we got some big things moving out. The nucleolus, a lot of students don't know about the nucleolus. The nucleus in a cell can have more than one nucleolus. It has one nucleus, but you could have 2 or 3 nucleoli, smaller little concentrations that look like a little sphere inside the nucleus. The nucleolus is where ribosomes are made. So we're going to be making organelles inside the nucleus. So not only do we have the DNA in here, but we're also making the ribosomes, which are another really important organelle. So inside that nucleus, we have chromosomes. Now, chromosomes, when we talk about chromosomes, chromosomes are DNA that's all wound up. Okay. And with it being all wound up, it's nice and we call it condensed. It's like me having your lab book. Okay, if my lab book is closed, I cannot access the information in there. So really the only time we close up our DNA, we close up shop, is when we're undergoing cell division, because the job of the cell at that point is to focus. It's like me telling my freshmen to close their Chromebooks and listen to me, focus. Okay, put their phones away, put your computers away and listen to me. All right, we're going to focus on dividing the cell. Okay, that's all we're going to do. But when the cell is doing its normal job, it's going to open up its DNA, and you can see that chromosome here unwinding. And it's going to open up unwound DNA is called chromatin. And we have these proteins that are going to actually wrap around. It's kind of like the core on your toilet paper, right? It's wrapped around some. So we have these proteins that the DNA wraps around to keep it nice and organized. So those are called histones. Chromosome number does vary by organism. Does not mean the more chromosomes you have, the more complex, more superior you are. There are some like yeast plants that have like 70 some chromosomes, right? Just because they have more chromosomes doesn't mean anything. They could be little tiny chromosomes versus bigger chromosomes. All right, so really when we look at the sophistication of an organism, we would look more like how many genes does it have, not how many chromosomes. The endoplasmic reticulum is a series of interconnected membranous sacs. We have 2 parts. We have the rough ER, which often I will call it the RER, the rough endoplasmic reticulum. Okay, you see it looks rough. It has these red little bumps on it. Those are actually ribosomes. And then we have the smooth ER, and the smooth does not have the ribosomes. The inner portion of the ER is referred to as the lumen. And you can see that the endoplasmic reticulum grows right out of the nuclear membrane. So they're not just connected, it's kind of an outgrowth. Okay? And you can see it with these different types of electron microscope pictures. Now, new proteins are modified in the lumen on the inside of the rough endoplasmic reticulum. So you have these organelles called ribosomes. Ribosomes are going to make proteins. As the ribosome, they start off free meaning they're floating in the cytoplasm. As the ribosome starts reading the instructions, there are instructions to tell it to stay free or you're attached. If it's attached, it needs to attach and hook up with the rough endoplasmic reticulum. And as the protein's made, it's inserted into the lumen inside here where it can be nice and protected and it can fold properly and be made the way it needs to be made. If it was made out here because of the water, it would not be made properly. Right. So we also know that the rough endoplasmic reticulum makes phospholipids. And actually, that is not correct. That is a mistake there. The smooth, so you guys might want to check that. I'm going to go ahead and here. Oh, let me do this. That should be the SCR. The SCR makes phospholipids for cell membranes. Okay. And that's going to be here, the smooth endoplasmic reticulum. All right. So moving to, let's see right here, lipids, makes lipids. Moving to the smooth, the SCR, there are no ribosomes attached to it. So again, that's why it looks smooth. It has a couple jobs. One, it makes carbohydrates, it makes some sugar, it makes lipids, makes cell membranes, it also makes steroids. Another thing that the smooth ER does is it detoxifies medications and poisons. And it also stores calcium. So it has a lot of different responsibilities. Now, when we talk about cells inside your body, we know you guys know there's a difference. There's clearly a difference between a heart cell and your skin cell, or a lung cell, or a cell in your small intestines. Those cells are all very different. They all have nuclei. They're all going to have ribosomes. They're all going to have rough ER and smooth ER. Now, the number of these in those cells may change. So, for example, the liver, your liver's job is to what? Detoxify you, right? You know, that's what cleans out your blood of any toxins. So when you look at the cell of a liver, off of a liver, it's going to have a huge amount of smooth endoplasmic reticulum inside of it. So it can do its job. When you look at one of your glands, like your pituitary gland, you're going to have a lot of smooth ER because, again, it's going to make hormones. When you look at muscle cells, your muscles require calcium for muscle contraction. So they're not only are they going to be full of smooth ER, but they're actually going to have modified smooth ER. When you take anatomy and physiology, it'll be so modified, you're going to call it the sarcoplasmic reticulum. So again, depending on the cell and its job will depend on the number of these organelles or how much of those organelles they have in each cell. Now, the Golgi is the only organelle that is capitalized. It is capitalized because it is named after the scientist who discovered it. So again, we have our proteins being made on the rough endoplasmic reticulum. They're nice and protected. We're then going to make this transport vesicle and take that protein that we made, and we're going to send it to the Golgi. And the Golgi is going to modify it. So I think of it this way. Your hamburger, the hamburger patty just got made. Your protein just got made over here. It then got sent to the topping bar. And over here, this is where you're going to put the lettuce, the onions, the ketchup, mustard, mayo, whatever you want on it. Of course, it's not whatever you want. It's whatever the instructions tells it. But this is where our proteins get further modified. Once they get modified and sorted out, because this is also referred to as the distribution center, because it tells it, okay, does it stay inside the cell? Does it go out of the cell? Is it going to be made into a lysosome? Golgi apparati also make other organelles called lysosomes. So all of these things are happening in the Golgi. A lot of different activity. Like I had mentioned, the lysosomes. So the lysosomes, when they are made up of a membrane. Inside that membrane, we have digestive enzymes. Oh, excuse me. Those digestive enzymes require an acidic environment. So the pH inside of these lysosomes are very low. It's like your stomach. Your stomach is acidic and you have enzymes that will only work in your stomach. So as soon as they go into the small intestine and the acid is neutralized by the pancreas, those enzymes will not work anymore. So this is a safety net here. If your lysosome starts leaking, you won't start digesting your cell, just like if your stomach starts leaking. So what we see here, we have this organism here. It has swallowed a bacterial cell. It is made of vesicle, so it's membrane. We have our lysosome, which also membrane. If you've ever blown bubbles and taken two bubbles and they've run into each other and they coalesce into a bigger bubble, that's what's going to happen here. If you haven't done that, you know, like you've shaken up oil and water and you've seen the bubbles and they come together and make a big glob eventually, right? And just all floats to the top. So this is what happens. They fuse together. And now you have those digestive enzymes that will break down whatever it's going to eat, whether in this case it's a bacterial cell. If this was a white blood cell, that's what they would do. If it is inside your cell, maybe you took in some food and it's breaking down food particles. It will keep what it needs, and it will get rid of the waste. So the waste could be anything from, maybe it is, I hate to talk about waste in details because again, you don't need to know about urea, or if we talk about maybe in some cases, maybe this is pectin or cellulose, other things that it can't break down. So we're just going to refer to it as nutrients. It's not even nutrients. Again, if those nutrients would keep it, so that was a bad word choice. So other than use, I can't think of a better word than waste. Okay. It's just materials that we cannot use. Okay. Vesicles and vacuoles are both membrane-bound sacs. Both function in transport and storage. So we're going to talk about, usually vesicles are smaller and vacuoles are much bigger. We're going to talk about this huge vacuole that we see in a plant cell when we get to 4.5. So like I said, there are very subtle differences other than size. I talked about the ribosomes already. So here's the ribosomes. It's actually two parts, a small part and a big part that comes and snaps together on what's called messenger RNA, which is a set of instructions that were sent from the DNA on how to make proteins. Like I said, all ribosomes start off free in the cytosol. And then at some point, if they are instructed to, they will attach to the rough endoplasmic reticulum. You can see the growing, this is the growing peptide chain. If you're like, what the heck's a peptide? That's a protein. Peptide is a fancy word for a protein. You guys all know what proteins are. The mitochondria and chloroplasts, I'll tell you right now, those are probably my favorite organelles, not for reasons that you probably understand. Most of my freshmen at the high school, when I would say, well, what's the mitochondria? They always get excited. They're like, it's a powerhouse of the cell. I don't know if they get excited because they like that it's the powerhouse of the cell, or if they're excited because they just remember that one organelle and what it does. I don't know. But its job is to take in sugar and to convert sugar into a currency of energy that your cell can use. That currency is called ATP, or adenosine triphosphate. It is very interesting to know that mitochondria also have, well, let's start here. They have an outer membrane and an inner membrane. Now, these details, I don't expect you to know so much now. We will spend a whole chapter on the mitochondria, I promise you. So we have the outer membrane, the inner membrane. The inner membrane has all these folds, which help increase surface area. Inside here, this is called the matrix. Side note, one of my favorite science fiction trilogies. And inside the matrix are, we have DNA. So the mitochondria have their own DNA. They have their own set of ribosomes that are different than our ribosomes. So they are structurally a little bit different. So it's really kind of interesting. And they divide by binary fission. That is how bacteria divide. So they will divide and increase in numbers inside your cell as needed by just dividing like bacterial cells. And they move around inside your cells. It's kind of creepy. It's kind of like, well, I'll get to the real creepiness here when we talk about the endosymbiotic theory. All right, peroxisomes. Peroxisomes have this layer, again, they're a lipid bilayer. They only have a single membrane, where the lysosomes, when we talked about in most of the vacuoles and vesicles, have a double membrane. They break down fatty acids and amino acids here. Now, when we break down fatty acids, we store extra energy as fat, lipids. When we break them down, or amino acids, we often get some waste products. So there's that waste that we cannot use. And some of those wastes can end up being toxic to the cell. So peroxisomes will detoxify it. Some of these byproducts are hydrogen peroxide. We cannot use hydrogen peroxide. It is corrosive, as you know, and can do some nasty things to your cell. And so the peroxisomes will take care of that waste. All right, 4.5. We're moving along here. So all plant and animal cells have those organelles. Now we're going to talk about the diversity of them. So we're going to talk about, okay, basically what it's saying is, what do plant cells have that animal cells do not have? Okay, so like I said, all eukaryotic cells have membrane-bound nucleus and many membrane-bound organelles. They are all enclosed by the plasma membrane, have genetic material, contain ribosomes. Ribosomes make proteins. If you're not sure what synthesize means, it means to make. Although there are many similarities, there are some important differences. So it's kind of cool because we're going to talk a little bit about those kingdoms. We're going to throw those kingdoms back: Protista, fungi, plantae, and animalia. So we're going to talk a little bit about the other kingdoms, which really gets me off track because I love studying kingdom Protista and fungi. It's kind of creepy, but I do. So I know I'm a total geek. I get that. I know that. I'm wearing a Tron t-shirt, for God's sake. Okay. So anyways, some of you guys are too young to even go, what is Tron? Google it. All right. 100,000 species are in kingdom Protista. Most are microscopic. Even though most are microscopic, some are huge and are like kelp. Seaweed is in kingdom Protista. Chrysophyta, phylum Chrysophyta. All right, we have, I could go on. We have phylum Chrysophyta, we have phylum Phaeophyta, we have phylum Rhodophyta. Okay, like I said, I could just go to town on this. This is like one of my favorite things to study. So they have a bunch of different types of organelles. For example, this paramecium here. Most of these organisms, many of these organisms in Protista live in fresh water. And so with living in fresh water, we're going to learn about tonicity and way water moves based on solute concentration. But basically, we're going to say there are more particles inside the cell than out. So water wants to come in and dilute out those particles inside the cell. It's like your basement in a flood. The water wants to come in. And so what do we all have in our basements? We have some pumps. And you can hear it when we have those storms pumping that water out. These creatures here, these little single cell organisms, also have some pumps. They're called water contractile vacuoles. And so water will constantly be coming in and they will pump the water out. Because remember, we don't have, these are very, I hate to use the word primitive. I'd rather use the word simple, single cell organisms. So they don't have organs. Like a fish would have an extensive set of kidneys to do that job. It's multicellular, but these are single cellular. So they have to have little organs, right? Organelles. So it will have a water contractile vacuole that pumps the water out. Fungi. This is where I really geek out. Okay, so there are different phylums. Phylums are under the kingdom. So kind of like we're going to subdivide. We got all these are fungi, and then we're going to divide them up into just some other types of groups. And we actually divide fungi up based on their reproductive structures. So you guys are most familiar with the mushrooms, right? Those are called the cap. Okay, so you have that mushroom cap. That would be phylum Basidiomycota. Then we have these groups that make this cup. The cup-like fungi, those Ascomycota. And then you guys all hate that. I hate that when I go to get my bread out and it's all covered in mold. That's your Zygomycota. And then we have some water molds, which is Oomycota. So they have all kinds of different variety things. And one of the biggest things is they have cell walls. And we haven't really talked about cell walls. Some of these have cell walls too, and those cell walls are made up of often pectin. Again, I'm not going to ask some tests, just a little side note here. And these cell walls are made up of different types of material too, often called chitin. So, and even though they have cell walls, so bacterial cells have cell walls also, it's made up of a sugar called peptidoglycan. So they may have cell walls, but their structures are different. They have the same function, but they're made differently because they're not closely related. So we do have a variety of different types of organelles inside them. But the main one that we are going to study is Animalia versus Plantae. And so this picture, which is funny, I think I have this picture on Canvas too, shows you all the organelles, the differences between them. And you can see they kind of box the ones that are different. We have plastids. Plastids are also, chloroplasts are actually type of plastid. Central vacuole, cell wall, and plasmodesmata. So we're going to focus on talking about them. We already talked about the centrosome and the lysosome. So we're going to focus on these organelles that plant cells have and animal cells do not. So plant cells have cell walls. So of course, most of our animals, right, we don't need cell walls. We have bones, right? We have a way to support ourselves. Plants do not have bones. You've never had to debone your lettuce before you make your salad. Right? So they have to have a way of standing up. You know, you got trees 100+ feet tall. How do they stand up without bones? They have cell walls. They're very rigid. They're made up of cellulose, which is for the most part kind of like paper, or we even reinforce that with lignin, and it's wood. I already mentioned that prokaryotes, their cell walls are made up of peptidoglycan, which is a sugar. Plant cells have chloroplasts. Chloroplasts are very similar to mitochondria. They have an outer membrane and inner membrane. They don't have the cristae. You could almost say that the cristae got so convoluted or folded, they pinched off. They totally pinched off from the inner membrane and formed these stacks called grana. Okay. Inside the grana is the lumen. We have the stroma. The stroma is the liquid in here. We also know that chloroplasts have DNA. Let me see if that's on the next one. No. They have their own, oh, it's right here. They have their own DNA. So they have their own DNA and their own ribosomes in here, just like mitochondria. And they reproduce by binary fission. Go figure. Oh, you heard me talk about this. Endosymbiotic theory. Endo, like inside, sim, with life. Okay. If we break down all those Latin roots there. So it is a relationship where one organism lives inside the other. So we know, like, for instance, I know this is kind of gross. You know, you have bacteria that live in your colon. We could call it the gut flora. Everybody talks about probiotics. You guys do know some of that bacteria comes out when you poop. Matter of fact, a lot of your poop is bacteria. Okay. So for the most part, they're there to help us. They help break down the waste that we cannot break down, and they actually also give us vitamins. So we have this relationship with those probiotics. They help us remain healthy, and we give them a home and food. So it's a win-win. Both organisms are getting something. Well, this is kind of the same with mitochondria and chloroplasts. They have their own ribosomes. They have their own DNA. They have two membranes around them. They're very similar in size to bacteria. They reproduce like bacteria, kind of like what's that saying? If it looks like a duck, if it walks like a duck, and it sounds like a duck, it's probably a duck. Well, it looks like a bacterial cell. It has the same structures as a bacterial cell. It divides like a bacterial cell. Maybe it was a bacterial cell. And so what we have found when we go back some 3 billion years ago, when we had our first eukaryotic cell, so notice we didn't have all these compartments and special jobs in the cell, but we start to form that endomembrane system. And as we start forming that endomembrane system, at some point in time, we've got an issue. And I'm going to tell you right now, a lot of people don't, when we talk about mass extinctions, these little guys get totally ignored. This little bugger is photosynthetic. So we're talking about in water. Life began in water, right? So we have all these bacterial cells competing for nutrients. This guy had some kind of mutation that said, well, I don't need to compete with food. I'm just going to lay out and soak up the sun. Must be nice. That would be cool to be able to just go out every day and just lay out and soak up the sun, maybe have some beverages, whatever. But the problem is this guy, when he's soaking up the sun, and you guys know, what do plants give off? Oxygen. There was no free oxygen in early Earth's atmosphere or in the water. It didn't come with the planet. Neither did the water. If you look at the history of our planet, fascinating. We did not start off with a drop of water on us. But again, that's for a different lecture. If you take earth science, that would be great. Now, oxygen. As photosynthetic bacteria started kicking out oxygen into the water started to become saturated. Oxygen is corrosive. In fact, if there was oxygen on our planet to begin with, life would not have been able to start. We needed an anaerobic environment. Anaerobic means without oxygen. So as this oxygen started to increase, we had probably one of the first mass extinctions. Those bacterial cells that couldn't survive, did not have the adaptation, they died. Some were lucky enough to have an adaptation that not only let them do well, but they were able to take in oxygen and use it to burn sugar to make high amounts of ATP. This big old guy at some point took in the little anaerobic, the little aerobic means it uses oxygen, bacterial, probably with every intent to eat it, when for whatever reason, it wasn't able to digest it for some reason. And it worked better with it. Okay. It's not like it said, oh, I like you, I'm going to keep you. But no, it was more successful. Cells that had the little aerobic bacterial, they could now cope with oxygen, and they got food, ATP, from the little guys. So they got a home and food, and the big cell got protection from the air, the oxygen, and they got ATP. It was a win-win. This group was very successful with forming this symbiotic relationship with what we call now the mitochondria, or mitochondria is plural. These are our modern heterotrophic eukaryotes. So these are heterotrophic, or remember, organisms that eat other organisms. So these are most of our, we can say, all animal cells, fungi cells, protists, most of protists. Well, a second event happened. So this would be like winning the lottery twice. that little photosynthetic booger got into the cell. And now this cell was very successful because it could support itself. It didn't need anything. These little green guys soaked up the sun. They made sugar. The sugar would go to these guys. They'd break the sugar down with oxygen that these guys produced. And then they would make ATP, and they also kick out CO2, which then these little green guys could take and make more. So this was a self-sustaining system. This gives rise to the modern photosynthetic eukaryote or plants. We also do have some photosynthetic protists as well. So this is the endosymbiotic theory. I think it's awesome, but it's also creepy to think that inside your cells are bacterial cells that keep you alive, right? They give you your energy. And then we have something that's not as fascinating, but it is the central vacuole. So again, this is a huge space inside. Matter of fact, it pushes everything to the side in the center of the plant cell. This has many jobs. For one, it's like that air mattress you bought. As long as it's nice and full of air, it's nice, and we call it turgid or firm. So this big water vacuole pushes out on the cell walls and keeps the cell nice and firm. But if we don't have enough air, you guys know, it starts to droop and gets uncomfortable. And in this case, the plant starts to wilt. And so we become flaccid. So it does help to regulate the shape of the cell and to keep it nice and we call it turgid. Okay. There are also some things that it does. Remember, we said plant cells don't have lysosomes. Well, some of those digestive enzymes are here in the central vacuole. We can also store proteins and other types of materials in here. It also can store waste. It can store toxins. It can have toxins in there. If you've ever had the lovely experience of, like poison ivy, poison oak. It also in some situations, it also stores pigments, right? So lots of different functions of the central vacuole. And then the last thing we're going to talk about here, I think it's the last thing, are intercellular junctions. So intercellular junctions, when we talk about cells, okay, cells, we have multicellular organisms. If it's just one cell, we don't have to worry about staying attached. But if you're multicellular, you have to keep your cells together. It's not good to fall apart. And so there are different ways that we are going to do this. And so we're going to talk about these. We're going to talk about plant cells and animal cells, how they do it differently. Plant cells use what are called plasmodesmata. One of my favorite words to say, just kind of, I don't know, just rolls off. Plasmodesmata. And these are basically openings in the cell walls. Because remember, you have those rigid cell walls, so you can't get through it. So they have these tunnels between all the cells so that the fluid, the cytosol, notice that you put cytoplasm, should be the cytosol, that liquid, can move amongst the cells. And so we can pass hormones and food and waste from cell to cell. They're always open, and as long as you can fit through it, you can go. Thou shalt pass. This is a problem because if a plant gets infected by a bacterial cell or a virus, if it fits through, it can quickly infect the whole plant. So it's not the best solution to the problem, but it has survived now, quite a few years in plants. We're talking probably at least 1 billion years or more. All right. Tight junctions. Tight junctions are, I always use the analogy, a Ziploc bag. If you've ever had a Ziploc bag that is a pain in the butt and you cannot get it to zip, and then you're like, oh, you are forced to do the pinch thing, right? Pinch, 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 then zip. This is the pinch. I got the pinch, the pinch, pinch, pinch. What that does is it prevents materials from leaking in between the cells, so we don't get movement. So here, this would be like the inside of your gut, your small intestine. We'll have these tight junctions up here because your fluid, your food, and your liquids all here. And you do not want it to go between the cells. If it goes between the cells, it will leak out into your abdominal cavity eventually when it gets on this side. And that's called leaky gut syndrome. It could kill you. Not good. So this is one area that we will have these tight junctions. We have a lot of them that in internal organs that protect leakage. Okay, so just like your Ziploc bags, right? We don't want it to leak when we use those tight junctions. The next we have desmosomes. The analogy I use for those, and remember those were those intermediate filaments, is that buttons. Think of a button-up shirt, right? It's very flexible. It can move, right? And this is what when we want tissues that are moving, like muscles that are stretching, your bladder that's stretching. You don't want those cells to pull apart, but we want them to be able to stretch and move. So the desmosomes are like little buttons that connect. So we also have those desmosomes here in the small intestine, because small intestines are contracting and pushing the food along. So we don't want them to come unattached either. So we don't want fluid leaking through, and we don't want the cells to pull apart. And then we have gap junctions. Gap junctions are similar. Their functions are very much so like the plasmodesmata. Okay. So with the plasmodesmata, again, it allows things through, but gap junctions are a little bit different. They can be open or closed. There are signals that will open them. There are signals that will close them. And they are also tend to be very specific for what they will let in and out of the cell. Right. So sometimes they'll let specific ions through, certain nutrients. They work together and they have instructions as to what they can. So it's kind of like a security gate there, but it will allow substances to move between cells. It's a little safer. And then we have the extracellular matrix. So again, besides having those junctions, those three different types of junctions, we have, I already mentioned we had that cytoskeleton on the inside that helps to anchor we've got anchoring those organelles between the plasma membrane and the nuclear membrane. But on the outside, we have all of these proteins and sugars that coat the outside. It actually kind of like they're sticky and let the cells stick. All right. So animal cells are mostly made up of glycoproteins. Glycoproteins are proteins with sugars on it, and collagen. Collagen's another protein. Now, the plant extracellular matrix is mostly just sugar. Okay, your cellulose, lignin. We'll talk about, well, I don't know what the new book will talk about primary, secondary cell walls, but you can see here all this stuff that helps it stick together. Oh, and so the last slide here, basically the last two slides, I'm not going to read through all this. You guys can read through it. It's basically telling you what you should have gotten out of this chapter, what you should have understood through reading, through the lesson. Some of this you might have gotten in the lab. And then of course, her references and stuff. Okay. So at this point, I am going to